So I want to introduce our speaker. He is Kirill Veselkov. He is assistant professor of computational medicine at the Imperial College of London and the Yale School of Public Health. And he will present it today, this talk that will be focused on integrating the AI, trying to model cancer. So please, thank you, Kirill, for joining us.
Thank you so much, Tanya. Welcome, everybody. And it's an early morning. I have to say, if I were presenting in London, I would probably see less people. But it's a great pleasure to be here and visiting you, Tanya. We applied for a large joint collaborative project together. Hopefully, that will come into fruition, and we'll be working together very closely. Today, I'm going to talk about, Tanya asked me to talk about artificial intelligence, machine learning, specifically applied to interrogation of omics data sets with regard to precision nutrition and precision medicine. So I'm going, since Tanya told me that there are going to be a lot of medical doctors who are not necessarily computational scientists. So I'm going to give a very kind of high-level talk, but please free to interrupt me to clarify any points, and I'll be more than happy to answer them. I wanted to start that it's quite exciting actually that with the technological advances that we currently living longer than ever. Actually, compared to a couple of centuries ago, the average life expectancy increased about twice. But at the same time, with the aging population, we are having lots of health-related problems, including cancers, neurological disorders, and diabetes. And a couple of years ago, we were struck with the COVID-19 pandemic as well. So, but at the same time, we currently can measure human biology at unprecedented level, actually, with the rise of omics technologies, including genomics, metabolomics. They're currently generating massive amounts of data. By the projection, actually by 2025, genomics alone, including single-cell genomic, can overcome the big data generators such as YouTube and Twitter. So we need quite advanced computational tools to extract actionable insights from such vast amounts of data. And the first topic I wanted to discuss today is how we can use machine learning to discover food-based molecules with the potentially anti-cancer and anti-COVID-19 properties, because the food we eat contains thousands of molecules. It's very complex, and that's where AI can help. But before I start that, I just wanted to emphasize that unfortunately, according to the most recent consortia published in Lancet a couple of years ago, that poor dietary choices are responsible for about 20% of all deaths. And what it's been found that it's not necessarily the junk food that kills us, but it's lack of healthy foods in our diet that are also responsible for a lot of death. And when it comes to cancers, by different estimates, again, they vary, but on average, about 4 out of 10 cancers can be prevented by dietary and lifestyle choices alone. So we need to pay a lot of attention to that. But as I said, that the food is very complex, and nutrition science has made an excellent progress in understanding 6 major nutrient categories, such as proteins, carbohydrates, lipids, minerals, and vitamins. But at the same time, there are thousands of other molecules. And it's also with the recent technologies such as mass spectrometry, we actually can measure these molecules in the food that we eat. So for example, it can be the molecule that's responsible for the dark hue of blueberries, or the unpleasant smell of garlic, or bitter taste of broccoli. So all this kind of chemical complexity is there, but we call it a kind of a dark matter, because it's not currently traced by national agencies. So when you buy, for example, food in the supermarket, you would see the mineral vitamin content, but you would never, for example, see the flavonoids that are present in there. So, but this is what we can currently measure with the analytical technologies. And that's why we need computational tools to predict, are there any kind of benefits of these molecules? And when it comes to analytical technologies, I'll cover it a little bit later during my talk, but we actually created an infrastructure, global infrastructure, to share mass spectrometry datasets in collaboration with the University of San Diego and the University of Connecticut in the United States. Our collaborators, Dr. Alexander Aksionov and Peter Dorstein, to basically allow everybody to deposit their mass spectrometry data and start sharing, and then we can try find the connections within the molecules, for example, we eat with the human blood, and start correlating this. This is another example. I will cover it later on, but I just wanted to emphasize that in order to be able to measure these molecules, we also need to create an infrastructure, and it's quite a complex way because you need to deconvolve simultaneously thousands of molecules in a single analytical run, and that's where AI can help as well. But coming back to the complexity of our diet, we swallow thousands of molecules, as I say, and they interact with our genome, with the guts, with the gut microbiome, and this system is ferociously difficult to analyze, I have to say. So let's consider a very simple example. If you take 3 out of 10,000 molecules, that would lead to about 1 trillion possibilities. If you were to use experimental methods, it would be like riding a bike to explore the galaxy. You will never get there. So that's where computational simulations can help us to distill some of the useful combinations of the molecules. And for that, we need a huge computing power. And currently, smartphone in your pocket that you have is actually more powerful than NASA computers that put the man on the moon. We didn't have supercomputers at that time, but nevertheless, the mobile phone is more powerful than the NASA computers that put the man on the moon. And currently, this power sits idle. You're not using it overnight. And jointly in collaboration with the Vodafone Foundation, we integrated the machine learning tools into the DreamLab app designed by Vodafone Foundation to harness the processing power of 1,000 smartphones to crunch this data on combinatorics of different molecules. So if you combine 10,000 phones in a single, let's say, a grid or high-performance computer, then it actually, you can do the calculation that we're currently doing in a couple of months' time. But if you were to do this calculation on a desktop PC, it would have taken us about 300 years actually to do such calculations. So this gives you the power of the smartphones. And you can actually download the app, and I would encourage you to do. It's very easy. You download it, click power DreamLab when you charge it overnight. And then during that, it will crunch through some of the combinations of the molecules that I just highlighted in the food and existing drugs as well, and they tailor it to specific genotype when it comes to cancer. And there are certain kind of limits on the system because the big data set needs to be divided into small chunks because the jobs needs to be independent because of the security reasons, and then the results needs to be aggregated. So the problem needs to be divided into small subproblems, each solved independently, and then the result would need to be sent back to us for the analysis. And I just wanted to show you a promo that we had when the project was launched a couple of years ago. And this is John Boyega from the movie Star Wars. And he came to our lab. I just told to Tanya and asked us about the project. And that video actually at that time we recorded when the project was launched to encourage people actually to use the smartphone. So I'm going to play it.
So I'm guessing I play a superhero in it. Flying, gliding, all that stuff. Oh, an ordinary guy. An ordinary guy, you know what? I like it. An ordinary guy fighting a one-man war against enemies from space. Against cancer. Okay, I see it. It's more emotional. It's more in-depth. This is my moment to shine. This is my inner actor. I'm just gonna use my phone. One man, one goal, one phone. I like it. I'm gonna be a sidekick to an app that helps to fight against cancer while I sleep. You know what? I'm in. Oh, I'm in. Hey, I'm John Boy. I'm John Boy.
Yeah, thanks to John at the time. But now back to science and what we're working on. We're working with the so-called molecular networks or graph structure data because inside our body we have a lot of signaling and metabolic processes that are governed by protein-protein interaction, and this is a protein in the so-called interactome network that they capture the interactions of about 20,000 proteins encoding in our genome and a couple of million connections between them. So if you take a molecule, it actually target specific proteins in this network, but these proteins don't exist in isolation. They're part of a larger network, and they have ripple effect on other proteins inside this network. And you can use so-called graph deep learning tools or graph propagation tools to perform simulation, actually, how that specific molecules interact with the human biological network, as I said, that govern a lot of signaling and metabolic processes. Of course, this is a much simplified process. It's a simplified process for a reason, because the system is very uncertain. And then you can take, for example, anti-cancer therapeutics and learn the pathways and molecular subnetworks that drive that therapeutics. And in this case, we built a machine learning classifier that would separate the existing anti-cancer drug versus all other therapeutics, and they built a machine learning model that will accurately predicting the anti-cancer therapeutics with the classification accuracy of 90%. So 9 out of 10 drugs were correctly predicted based on their interactions with the molecular networks. And this is the network highlighting the top 10 pathways that we learned from the existing therapeutics to accurately classify it. And you can see that this is kind of the hallmarks of the cancer, multi-stage cancer development. And once we've done it and kind of validated the model, we then process thousands of food molecules through this model to identify the molecules with the potentially anti-cancer properties. And we have discovered about 110 molecules, and this is basically a summary of them. They're coming from different classes, like indoles, for example, flavonoids, terpenoids, and this is quite a chemically diverse classes, because we focus not on molecular structure, but on their biological effects on molecular networks. What's really exciting that it gives a lot of probably headache for the future regulators, because as I mentioned, if you currently summarize all the components that are currently traced by national agencies, for example, such as United States Department of Agriculture, there would be 145 plus or minus components that are currently traced, including all minerals and vitamins. And here, for example, we have discovered 110 additional ones. So how would you take those measurements into account if you want to establish the kind of the health related effects? So that's really, I think, big challenge in the future. And in classical kind of way, you would go ahead and do lots of experiments, probably elucidating the mechanism each of these molecules. Although, as I said, that we have predicted some of them using graph-based tools, but in order to validate our discoveries, we also developed the natural language processing engine to extract that information from the literature so we can back it up with the simulation that we perform to validate our discoveries. And actually, it's quite funny, but in the field of cancer, there is about one paper published every 10 to 15 minutes, approximately. So by the time I actually complete my talk, there would be probably 2 or 3 new publications in the field of cancer. And in order to keep up with the this massive amount of information, you actually need natural language processing tool and artificial intelligence tools to extract that information from the literature. And I think in the future, what scientists need to think about how to make papers not human readable, because when you give it to even 3 independent experts, human experts, they can interpret most of the time, probably findings quite differently, just looking at the same paper, but how to make the manuscript machine readable, actually, so we can extract the information in automated fashion, given the large number of publications. But as I said, that as part of that, we have discovered 110 components that we have cross-validated with the experimental literature, with the experimental data, extracted from the literature. And then we've taken these components and mapped them using the mass spectrometry data I mentioned before into different ingredients. And here you can see the food map, where the size of the node is proportional to the diversity of the anti-cancer molecules that we have discovered. You can think about whether kind of enrichment score with regard to the components that we have discovered. So the higher, the more diverse the molecules it has. Of course, there are lots of different things you need to take into account. For example, I don't know, like cabbage grown organically in your garden versus the one that you buy in supermarkets probably would make a huge difference in terms of the molecular composition. And I think that's how it's grown, how it is cultivated, how it's processed. All these parameters need to be taken into account. And of course, it would impact not only the composition, but also the bioavailability of these molecules. But if you take a purely theoretical view and take existing data, that's what we would have. I think what's really interesting to see, although some of the obviously ingredients have more enrichment in terms of anti-cancer molecules, but more or less every ingredient has some interesting phytochemistry in it. And rather than focus on one individual ingredient, although I do start my mornings now with cabbage smoothies, but it does taste quite awful, I have to say. But rather focus holistically on a diet and basically having phytochemically enriched diet versus controlled diets may be the way forward, because you can substitute some of these ingredients, as you can see, because they're currently clustered and color-coded according to the similarity of their chemical composition. And so this is some of the comments that we get. I mean, that's from Eric Topol, who is a pioneer of digital medicine. But you always have people who say that you cannot trick me in eating more fruits and vegetables. So that's the way it is. And when it comes to combinations of the molecules, the way we've tailored our system, we've also incorporated a lot of data from the drug synergy studies, and we've trained the model to predict the synergy between the molecules. Of course, this is a pairwise, again, very simplified way that was done by Guada, who is a member of my team. And the same can be done with regard to the COVID as well, because when it comes to SARS-CoV-2, it actually uses our cellular machinery to replicate and to survive, and resulting in a virus host interactome networks. Virus itself has 30 plus proteins approximately, but interact with a couple of hundred proteins inside our cells to replicate and to survive. So you can imagine that forming that interactome network with the disrupted pathways, amended metabolic activities as well, and that can be targeted by the molecule. So you can, on one hand, propagate how the virus interact with the cell, simulate that process, and on the other hand, take the molecules, for example, perform simulations and find the molecule that would target that virus host interactome network. And especially with the potential escape of the vaccines, that can be one of the ways to, for example, prevent the severity of COVID-19, but also we currently just about to launch the project on long COVID as well, to kind of modulate the pathway that are responsible for the symptoms associated with the long COVID as well. And that's exactly what we did. And here we have discovered 45 components that target specifically virus host interactome networks, say lead candidates, and then we map those into ingredients as we did, and created a food map that you can see on the picture. When from our theoretical calculations, I have to say that things like, for example, blueberries, black currants, so lots of berries, for example, broccoli, cabbage, they have quite phytochemically rich profile that target this virus host interactome networks. And we ideally would like to do the clinical trial to have, on one hand, phytochemically enriched diet versus the control diet, and to see what the actual effect it has. But from the studies that was published by Tim Spector, who is at King's College and doing lots of research on precision nutrition, they found that plant-enriched diet reduced quite substantially. They published a paper in, I think, in the Gut journal, quite substantially reduced the risk of developing a severe COVID related symptoms. So that's at least some theoretical backup why that can be potentially the case in terms of molecular biochemistry. And of course, as I said, that my cabbage smoothie tastes quite awful, and a good friend of ours and my collaborator, a molecular gastronomy chef, Joseph Yusuf, who I met a couple of years ago when I was giving a talk at the conference on the future of computing and food. And we spent with him 3 days in Italy discussing intersection between the computational science and food science. And they had quite a few bottles of wine in Italy. And that's how he got involved in our project. And here he's doing kimchi, the traditional Korean fermentation dish, 250 kilos actually of kimchi that we gave at one of the festivals that we had at Imperial College just before the pandemic actually. So again, and fermentation, we just had a study carried out in collaboration with the University of Connecticut, where we have measured actually different days how the fermentation process changes the chemical composition as kimchi kind of matures, which is very interesting because lots of molecules pops up, lots of molecules kind of disappear. So there is a whole new angle actually of how to cook the food and prepare it. Regarding the cooking of food, again, where the graph machine learning can help us a lot, because you can represent recipe as a computational graph. So you have inputs, you have different processes, you're cutting, for example, chopping, boiling, and it's forming this very large graph. You have on top of it the so-called the flavor pairing network, because you want to maximize the flavor, the taste of the food that you eat, but at the same time, you also want to maximize the health benefits, which can be in contradiction to each other. So let me give one example. So for example, the cabbage that you buy in the supermarket, if it has a bitter taste, it may likely to have more indole carbonyl, for example, compounds that is responsible for those tastes, but that may not necessarily be good for the consumers who prefer more sweet taste of cabbage. So that can be a dilemma actually, how to do two things. One kind of health benefits, the other one is a taste profile. But nevertheless, and this is a part of a very exciting field that is just at the beginning. It's a computational creativity, because create, for example, recipe is a computationally creative process in itself. Probably one day we can have a competition between the kind of AI generated recipes and kind of molecular gastronomy chef and see which one is better. But in the meantime, we've relied on Joseph's experience, and we published a kind of hyperfoods cookbook based on the ingredients that were discovered. And so this is for long COVID, and you can download it and take some of the recipes. I have to say that my wife says that these recipes are quite complex, so take your time to prepare it, but it's an artist view of the discoveries. I have to say that don't use it for medical purposes. It's an artist view of how to present basically the science to a general public rather than use it as a prescription. But coming back to the kind of the technological angle, why the molecule that we measure in food is possible, as I mentioned before, that's largely possible due to the developments of analytical technology such as mass spectrometry that enables to measure thousands of molecules in the food, be it in the blood we collect from our patients, in the air even we breathe, in the water we drink as well. So this is very complex chemistry, and compared to genomics data, where from the decoding of the human genome, we know the proteins composition, for example. Here, the diversity of small molecules is very large, but when it comes to clinic, they're very important because I think about 90% of current diagnostic and prognostic tools are based on small molecules. About 90% of all drugs are actually small molecules as well. So, and also metabolites are product of gene environment interactions as well. So they are not only influenced by our genome, but also our gut microbiome and exposome as well. So they're really important molecules to study. But when we do study them using mass spectrometry, I have to say that there are thousands of molecules colluding, and it's a big problem to deconvolve them and to get reliable measurements, and very difficult to scale up. So before we started working on it, I mean, it was a really difficult problem, and the data usually published as a supplementary kind of to the manuscripts, and then you have to, and they just buried actually in that large bibliographic kind of databases, and therefore that what facilitated us to develop an infrastructure, contribute in the infrastructures that ultimately led to the sharing of these data sets. And that was done, as I mentioned, with the University of San Diego and Connecticut with our collaborators, and we published it last year, and develop a MS Hub solution that capable of deconvolving these molecules at a repository scale, basically hundreds of thousands of spectra, or even millions of spectra that aggregated and contributed from all over the world. And once they're deposited and processed, there were a lot of computational challenges that we needed to overcome, which you can read in our paper that was published in Nature Biotechnology. I would encourage you to do so. But we used it to process various types of data, including human blood samples, urine, food, exposome, air, and even human skin, for example, is through the patches. And once they are processed, then you can start correlating these data sets and see, for example, if the molecule that you found in food, where it also links to whether it's found in human blood as well. So you can start doing inferences that wasn't possible before when the studies were done in isolation. And so that's comes to one of the applications that we've also delivered, one of the use cases of the platform, since it's main interest of Tanya as well. So for the diagnosis of early gastric cancer, because in the human breast, the cancer cells inside our stomach, they produce quite a distinct volatile signature because of the biochemistry or the interaction with the gut microbiome that we breathe out, and you can collect human breath and using so-called gas chromatography mass spectrometry, you can measure volatile organic compounds that they partially originate from the cancer cells. And that signature you can use to distinguish, for example, early gastric cancers versus controls. And here, for example, with the area under the curve of 0.9, we were able to discriminate, again, using graph molecular networks, the graph or motifs, bless you, the motifs that would discriminate early cancer versus control. And we currently, as part of CIUK program, we are actually doing a lot of biological experiments to understand the specific origin of the volatile signature that we have discovered, and to scale up to 6,000 people, actually, the clinical trial that we're currently doing. But very exciting kind of technology, but fundamentally, you need to deconvolve these molecules because you need to know for the diagnostic tests and for explainability what these molecules are. And in this case, we are not using the levels of the molecules directly, but rather than the molecular subnetworks to classify early gastric cancers. You can measure that molecules on human skin as well, and that was done in California. So you have quite a bit of sunscreen, or basically the molecules from the sunscreen that was put on the body. And again, I mean, that's the biochemistry that we exposed to, but we fully understand what it's doing to our body. So lots of exciting things. You can also measure these molecules in the habitat, basically in the place you live, and also see whether you have some dangerous molecules in your place itself as well. That's currently done, obviously, for scientific projects right now, but maybe in the future that can be kind of envisioned, that you have a couple of sensors that can measure these molecules. Well, and to conclude kind of my talk on, if I still have time, maybe a couple of minutes, because Tanya asked me as well to focus on digital pathology a little bit, and I just wanted to show the platform that we delivered specifically for mass spectrometry imaging that basically allows to measure thousands of molecules, but up to cellular resolution, basically get this complex profile to study, for example, heterogeneity of cancer or tumor microenvironment. But also it has a potential application in digital pathology, because currently the tests are done by qualified pathologists who take basically the slides and look at the high resolution, I have to say, microscopes, and interpret whether you have cancer or not, what type of cancer, obviously, and different characteristics of cancer, and also with advanced staining assays perform some molecular characterization of the cancer. The problem with that approach is that if you give the same slide to 3 independent pathologists, when it comes to different difficult type of cancer, that can be interpreted quite differently. So there is this inconsistency. So the mass spectrometry imaging basically can measure the chemical fingerprint of the cancer cells. And you can use that chemistry actually to classify and understand the cancer, because obviously morphology is an important characteristics, but it's one of many. Ultimately, we need to understand at chemical level what's going on. And mass spectrometry basically allows you to measure the mass spectra I just showed, but at spatial resolution with some technologies actually up to 1 micron, basically going into individual cells. That generate quite a huge amount of data, I have to say. But as an example, I just wanted to show you how the machine learning can be built to accurately classify different types of cancer using the chemical signature rather than morphology. And for that, we will, an example, show the supervised learning procedure. And here, what we have is colorectal adenocarcinoma, colorectal adenocarcinoma islands. You can see with the blood vessels embedded and surrounded stromal and muscle tissue. That's quite a nasty one, actually. And so what we're going to do, we're going to select a couple of regions. So here in red, it's cancer tissue, green blood vessel, and blue muscle tissue. And I'm just annotating them. In a second, we will kind of real time build a simple mathematical model that would discriminate the tumor versus other tissues and also the blood vessels and muscle. So here, even from principal kind of, in this case, unsupervised learning principal component analysis, you can see that the chemically, these three regions are quite different. And here we can get the idea what kind of molecules contributing into this separation between these three different regions of interest. And I'm just zooming in. This is was acquired using so-called desorption electrostrainization. So it mainly covers fatty acids and lipid composition inside the cells. And this is just one of the potential biomarker with mass to charge ratio for 309. So probably one of the fatty acids that discriminate that is different in terms of the levels between the tumor blood vessel and muscle tissue. And of course, I mean, that's currently done manually, but we can use machine learning tools to find the signature that would discriminate in mathematically optimal way tumor, for example, versus blood vessels and versus muscle tissue. And we'll do it in a second and try to reconstruct actually the whole slide from these signatures. But this is just showing some of the kind of data, how they look like after processing. And this is obviously the molecules that we annotated from that specific slide. But I wanted to show you how it looked like. So we're basically taking these examples from that regions of interest. We've built mathematical model that discriminate or find the signature that discriminate that region of interest, in that case, colorectal adenocarcinoma cells versus blood versus muscle tissue on that specific slide, and derive a signature that would accurately predict that specific regions of interest. So once that signature was derived, we then apply the model to predict where on that slide there are other colorectal cancer cells, and here basically in red you can see that it you can trust me on that one, that it quite accurately matches the morphology that we have on morphological staining. And the same we can do for the blood vessel, and the same we can do for muscle tissue. And then using the chemical information that we learned from that slide, we can accurately reconstruct all three components specifically on that slide. Of course, to build robust model, we will need hundreds, if not thousands, of examples to derive the signature, because it varies according to not only the analytical instruments, but individual characteristics. But nevertheless, that's something that just an example how the machine learning can automate this process. But in addition to the kind of morphological diagnostic, you get hundreds, if not thousands, of other molecules that you can map into pathways and go deep into understanding the molecular insights, for example, about the cancer itself, about the tumor microenvironment interaction, because from those kind of other regions you can extract as well. And what we found, for example, that stroma sometimes is more predictive, stromal composition is more predictive about the cancer response than the cancer itself, for example. So all these nuances you can, because cancer actually uses its surrounding tissue to proliferate, so it changes it, it modulates it. So that's, you can use actually for that purpose. So this is an example, again, without machine learning tools, you cannot actually interrogate the data because they are quite, they're very, very large. And Again, I mean, this is a couple of papers that my team published, and we outsourced the platform actually to process such data sets. And feel free to download the respective articles. And one of the good things when you develop the platforms that if lots of people using it, then you first of all get lots of publications, but also it's a very similar workflow, but different types of cancer clinicians can use it and to address specific unmet medical needs for their specific use cases. But the good thing that underlying AI technology or machine learning technologies is very similar. But there are a lot of as well the chemoinformatics actually that goes into that kind of platforms, because you need to understand the physics of the instruments to be able to interrogate such data sets. And on the second example, I wanted to show just kind of unsupervised learning. And here we have basically hundreds of examples of colorectal adenocarcinoma that is here through the unsupervised learning, where we didn't provide any information to compute about the region of the tumor where it is located. It just learned based on the similarity of chemical profiles. And here it's differentiated quite well with the white colorectal adenocarcinoma versus stromal muscle and other tissue types that are highlighted in yellow. So this is an example where we don't have any kind of prior information about the tumor and use the chemical information and through the unsupervised learning to cluster similar regions in terms of the biochemistry that matched with the pathology quite well. On that note, I will conclude. I would like to thank my team because they've done most of the work, I have to say, and particularly Ivan, I think Dennis, Michael, Joseph, Guada, Lida, and many of my collaborators as well, Alex from United States and Peter, also Josh. And I would like to thank Tanya once again for inviting me to give a talk in here. If you have any questions, please feel free to ask.